So gene expression is what we've been addressing here in this past couple of videos with DNA and RNA discussions. And it's been leading up to this very point. And so you see in this particular picture the idea that what we're going to do is we're going to take a short stretch of DNA, we're going to separate it apart. On one of those two strands of DNA contains the coding for the protein that we're looking to express, the gene, right? The gene is the coding. And this is happening within the nucleus. Now, once the strands of DNA are separated around where the gene is found at, we can begin to transcribe those bases that form that gene into the bases of RNA. This process is termed transcription, and it happens in the nucleus. Now, once the messenger RNA is created, it can exit the nucleus through a nuclear pore and enter the cytoplasm. And when it enters the cytoplasm, it will find a ribosome, making up some ribosomal RNA. And along with tRNA, then we can have translation of the codons of RNA into the amino acids making up the protein that the gene is coding for. So this is the central dogma, the major component of biology, if you will, that we have transcription that takes DNA to RNA. We're converting the 4 bases of DNA in the gene to 4 bases of RNA. Not a big change there, so we're basically transcribing, just like a secretary transcribes a boss's notes. Then the second half of the equation occurs in the cytoplasm after the RNA molecule has left, and that is the translation of the process. We're changing from a 4-letter alphabet in the RNA to the 20-letter alphabet, if you will, in the protein, right? The 20 amino acids. So that's the idea of gene expression, a broad overview of it. So there are certainly a lot more complicating factors here, and we can get into some really gory details about transcription and translation, but we're not. I know you'll be sad about that, but we'll just consider the basics and just know the overall scheme of things, that DNA is resident in the nucleus. The gene on the DNA molecule is converted to RNA. RNA is what leaves. It's converted through the process of transcription. It is RNA then that exits into the cytoplasm. So that's the process of transcription. If you wish to go over it in more details, I'd be happy to send you to websites that have more about it, or you can come into the office and we can certainly discuss it more. Translation, on the other hand, is the process of converting the bases in RNA to the 20 amino acids. And so we require the ribosomes. This is going to occur in the cytoplasm, and this is the specific process, the sequence of events. You see the messenger RNA that is present in the part coded with the red line. So we have the messenger RNA. It's attached to the small subunit of ribosomal RNA. And then what we have attaching to a particular codon is the tRNA that starts the process of building the protein. Now at this stage, the large subunit will dock and we'll start to have assembly of the protein. Again, I don't expect you to follow all of this in gory detail. I just want you to understand the very basics, that the process of building a protein involves messenger RNA attaching to the small subunit of ribosomal RNA, and a particular codon in the messenger RNA has a start code that the first tRNA will dock on. And then we'll begin with the next 3 letters. We'll slide over, we'll begin adding more and more and more amino acids onto the protein until we hit a stop sequence when the building of the protein will cease.